Welcome everyone to the sixth episode of MCC International Webinar Series. This is brought to you by the Mabalacat City College, Office of the Vice President for External Affairs, and the Institute of Arts, Sciences, and Teacher Education. Our guest speaker is a PhD student in bioinformatics at the Norwegian University of Science and Technology in Trondheim, Norway. She works with biological pathways and gene expression analysis in prostate cancer, and her main focus has been on improving pathway analysis by incorporating functional homologs and read counts from gene sequencing. The nodes and links of biological pathways can be altered with color or shape or size to show additional information about what's happening in the cell, like a still picture of a situation at a given time. She is the main creator behind FunHop, a tool which further improves these still pictures into a more biologically correct analysis. Her topic for today is on how FunHop changed the picture. Friends, let's all welcome Miss Jers Teresa.
Hello everyone, my name is Kjersti Riise, and I named the talk of the day how FunHop changed the picture. And hopefully I'll be able to tell you how FunHop actually changes the picture and why this is useful. I'd like to start off by thanking the organizers for all these beautiful talks. It's been very fun and educational so far, and I'm hoping it'll stay that way. So let's just get started. This is me. In 2015, I finished my master's degree in biotechnology, focusing on systems biology at the Norwegian University of Science and Technology, also known as NTNU, where I also started on my PhD in bioinformatics in 2015. And it's not quite finished yet, but I'm hoping we'll get there soon. So we're based in Trondheim, which is in the middle of Norway. It's a beautiful city with a big river and small houses, crooked streets. We have a big cathedral, northern lights during the winter, and snow, and it's very beautiful. And this is the main campus of NTNU. This is the outline for today's talk. I'll start by talking a bit about networks and just showing some basic properties, why they're useful, and how you can use them and what you can learn from them. I'll talk a bit about tools, like the databases you can choose from and the visualization tools you can use, and then I'll talk a bit about FunHop, a bit about how FunHop works and how it actually improves this picture by using two different examples. So, if we start about some useful features about networks, we see here a small network with only 5 nodes. So the circles here are nodes. They have different colors showing they're different. They are connected by links or edges. So we can see here, there's not a lot to read from this one. It's very binary. They're either connected or they're not. And the matrices here are similar on the diagonal, showing that it's not like the yellow one talks to the red one, but the red one doesn't talk to the yellow one. There's just a link between them. We don't really know a lot about it. So if we continue with the talking example and we add some directions to these, we can say that the red one talks to the green one. Actually, everyone talks to the green one, but the green one doesn't talk to anyone. And only the purple and the blue one actually talk to each other. Everyone else is just one way. And then, as we can see here, we actually add more information on these links. Like, for instance, the red one just talks a little bit to the green one and not to anyone else, while the purple one talks a lot to the green one. And these are some of the things that you can do. You can add informations to both the links and the nodes, and then you can see how sort of the network changes by adding these extra informations. There are also some other features in the network you can look for, like for instance a bottleneck or a hub. Hubs, for instance, would be very important because, as you can see in the little hub area here, if you remove the hub, you will actually lose a lot of the nodes from this network. Nodes M, Q, S, L, K, and P will actually be gone from the network if you remove node J. So these are just things you can look for when you're using networks. And of course, they're not usually as small as this one. We have some examples here, like the human disease network, where all the node side is proportional to the numbers of genes participating to the corresponding disorder. You have the disease gene network, where the different colors correspond to a disorder class. As you can see here, the nodes can have different shapes, different colors, and they can also, as you can see here, it continues with different shapes, different colors. It shows some form of information about the node or the link, and this is very useful. And of course, if you go up to a really high level, you have all the metabolic pathways. And I'm sure you've all been shown this at some point during biochemistry or another related area. And you can see the different forms of metabolism, how it's all connected while it's still remaining its own special field. So I'm just going to go quickly through some of the databases and visualization tools you can use to make these networks. There are obviously a lot of things to choose from here. There are databases and tools, and you basically have to figure out which ones you like to use, some of them you have to pay for, some are free. There are different features to all of them, and you just have to find out which one you prefer to use. Some of the ones that I've used as databases are Reactome, Panther, Wiki Pathways, and KEGG. FunUp uses KEGG. KEGG is manually curated, and that works fairly well. Wiki Pathways, for instance, can also have pathways made by users themselves, so you can have fresh ones, new ones, all kinds of stuff. You can just play around with it and look at them. One of the things that I like to do is download the tools from here and into these visualization tools. And there are many different types of these tools. For instance, you have Pathway Studio and Cell Designer and Cytoscape. And they all have different features, and you just have to find one that you like to use and basically just go ahead. Personally, I'm a big fan of Cytoscape, which is free and open source, and they have a lot of extra so-called apps that you can download, which can also expand which types of analysis you can use. And like FunHop, if you have a need that you feel is not fulfilled, you can actually make your own app, and it can then be shared with other Cytoscape users, and they can use it too. That is the main goal for FunUp sometime in the future. It's not quite there yet, but it'll get there. So let's look a bit into KEGG, or the Kyoto Encyclopedia of Genes and Genomes, which is a database of pathways. And as you can see here, they have a lot of stuff. They start, for instance, with metabolism. They have carbs and lipids, nucleotides, amino acids, special cases, basically everything you need. They also have genetic information like transcription, translation, folding, replication, environmental stuff with membranes and signals. They have cellular processes like transport, cell growth, cell death. Organismal systems, immune system, endocrine system, circulatory systems, nervous system, sensory system, all kinds of organismal systems. They also have a section on human diseases, like different types of cancer or immune diseases. And they also have a section on drug development. So this is fairly simple. You just open up KEGG on a website, find the one you want, and then if you click on it, like I did here with histidine metabolism, and choose human, it looks like this. So all the green ones are the ones that are found in human and the ones that are related to this pathway. And this is actually how FunHop came to life. When I was a fresh PhD student, I went to a lot of talks and saw a lot of medical doctors and also scientists who actually took screenshots of pathways like this one, also from other databases, of course, but they would just take a screenshot and add a little star to a gene they had found that was upregulated in its own form, and from that they would read a very lot of information into it and say that this would change the entire pathway. And well, as we all know, it's not really as simple as that. So that's how it all started. And if you take a pathway from KEGG and open it up in, for instance, Cytoscape, using one of the XML apps called, here I've used KEGGscape. It looks like this. And there you can see how the green ones are still green and they're just now in this tool. And you can see that there's a list below with information on the different nodes. And from here on we can start playing around with it. Like I said, there are many different apps you can use that will add all kinds of features to this network. And I'm going to show you how FunHop does it and how it works in the next. So this is the same pathway as before. As you can see, it's a little bit different. That's because this is the actual pictures from the article about FunHop, which hasn't yet been made available, but we're hoping it will be soon because it has been accepted. So this is the actual figure. It looks a little bit prettier than the one in Cytoscape, if I may say so myself. So what we can see here, it's still the pathway on histidine metabolism. But what we've done here is that we have log transform p-values from differential expression, where we took data from the Cancer Genome Atlas and we did differential expression between cancer and normal samples from prostate. And this is what I talk about when I say the picture. So this is a still picture showing the situations in this pathway when you compare cancer to normal cells. So what we have here in the differential expression, we see the little bar in the bottom left corner. So if one of the nodes are gray, it means that they're not significant at all. And if they are light yellow, it's close to 0. And then all kinds of green means upregulated, and to the strongest dark green bluish, which corresponds to a log transform p-value of 600, whereas all red, purple, and black tones are downregulated. So if we're gonna look a bit into this, there are two different sections within this pathway which I want to show you and I want you to pay close attention to. So we find histidine itself is here, and what we know from some metabolic studies is that histidine is upregulated in prostate cancer. So there are more histidine found in prostatic cancerous cells than in normal prostate cells. But if we just look at this picture here, is there any way to explain this? So if we, for instance, go from carnosine here, you see there are two arrows showing that the flow will go down here and down here. So this one is upregulated and this one is downregulated, and we don't actually have any information outside this to say, okay, so one of the genes that transforms carnosine into histidine is upregulated, one is downregulated, so does this explain why there's more histidine? And then again, you have this gene here, which is also upregulated, which transformed histidine back to carnosine. So none of these actually can explain what's happening. We also have an upregulation here. So does this mean that histidine goes to urokinase? We don't actually know because we don't have the numbers on urokinase. These are not significant, so we don't actually know what's happening here. So we can't really say or we can't even try to explain why there's an upregulation of histidine in prostate cancer. That's the first thing I want to talk about. The other thing is this path down here from histamine. Because as you can see here, this is downregulated, this is upregulated, and this is downregulated. Now you don't have to actually remember this because I'll show it to you later, but just notice it. That's how it works. And from here it might seem like, okay, why is this important? What are we doing here? And does this even make sense at all? And what is the problem? Which is perhaps the most important one. And now I'll show you the problem. Because now we're back in KEGG. And as you can see here, my little pointy hand basically means that I hold my mouse pointer above one of the nodes. And as you can see here, this is the path that I showed you from histamine downwards, where you could see only one gene at the time. But if you hover your mouse clicker above a gene, you suddenly see that there are 4 different genes involved. And we don't know anything about this other theory. We did see the ALDH3A1, which was the top one. We did see that one, but we didn't see anything about the other ones. So is it possible that there is more information found somewhere within these other genes that we simply cannot see? And that's the problem here. We didn't see them. The picture didn't show it. So if we were to analyze based only on the picture in my previous slide, you couldn't. But we have a solution to this. Because, for instance, one of the ways that KEGG makes, for instance, their protein complexes, it's like this. So a protein complex are proteins, different proteins have to be combined to work. And that's made by making a little box or a combination node with all of them in the same time. And this feature we can actually use. And we did. Because FunHop changes the one node with one gene into a node complex with all 4 genes. And as you can see here, you have all the 4 genes from above is now found within this box. So if we then apply this to the network we had, we do everything else the same. We have the same differential expression that's already been made, doesn't matter. We can apply the same colors for up and down regulated genes. We just show all the genes. And then we end up with a picture like this. And like I said, there are 2 things to look at. Up here, there weren't any other genes found, so we don't actually know anything about this area. We cannot say anything about histidine. But look at what happened down here. So remember we had just 3 single genes here, which are the ones on top. This one's still just 1, but this one suddenly has 2. One upregulated, the one we saw before, and suddenly there's one downregulated. And here are the 4 ones that we saw before on the KEGG website. And even though these 2 are downregulated, these 2 are upregulated. So we actually have no idea what's happening here. For instance, over here in this node, this top one was downregulated. All these are downregulated apart from this one, which is not significant. So nothing major happens here. But these 2 actually have different directions going on. So here's another problem. How do we know which one of these are actually doing the job here. So what we can say here is that these are functional homologs, so there's not enough data to say which one of these are the ones in this particular space doing this particular job. But what we can say is that since they are functional homologs, they work similarly, they do the same job, and we can therefore assume that they will do it on the same rate. And if we assume that, we can also assume that the one gene that is found in the highest read count is probably the one responsible for this particular transformation. So, how do we figure this out? Well, this data, the differential expression, is based on gene sequencing, because they've sequenced the cancer cell and the normal cells, and we have all the read counts from this. So if we take the same network as here, but we just apply other data to it, we see a different picture. And that's what we did. That's what FunHub does. So here, this looks different. They're different colors, but it's the same network, just showing different information. So if you look at the bar down here, you see that 0 is close to the end here, and it basically means that if something is white or really, really bright pink, it has a very low read count. And the bluer it gets, the higher the read count. And up here, you're close to 50,000. And so everything here is normalized against gene length. So you should be able to have a fairly decent read count number. And then let's start with this case up here, because it's easy to explain. There's not many things going on. We start here. We see here CNDP2. It's blue. It's somewhere around here. So it's a decent number of read counts. The exact number is not that important here, because what we can compare it with is this one. Remember, this was upregulated, this was downregulated. And this one is white. So there's close to 0. There's nothing. And this one, which transforms it back, is just really, really bright pink, which means there's very few of them. This sort of pushes carnosine down towards histidine. There's a high number of these. We can assume that this means that there's a lot of transformation from carnosine to histidine, which would give a high number of histidine, and it's not going back this way because there's not a lot of these. Well, these two are also fairly low, and this is close to 0. This means that we suddenly have a plausible explanation to why there's a lot of histidine, because this one pushes the flow this direction. And suddenly we can actually have an explanation to why this happens. And now if we go back to this area here, this one's just alone, so it doesn't matter. And what you can see in this node here, you can see that it's MAOB, which has the highest count over MAOA. Okay, what does that tell us? Well, if we look at it compared to this one, it seems like the upregulated one has fewer reads than the downregulated one. And this basically means that there's more of this than this. Okay, what does that say? In this box down here, we see that these two genes have a very low count. This has a bit higher, but it's still nothing compared to LADH1A3, which is close to 50,000. I think it's about 46,000, which basically means that it's this gene that is the dominant one in this entire node of genes. Okay, but is this useful? Can we learn anything from this? Is this not just complicated. I mean, looking at these, comparing, this is a small network, it's fairly simple to look at, but what happens if we have a big one? This would be a problem. But then again, FunHub comes in and saves the day, because what FunHub does next is that we don't look at differential expression on a gene level anymore, we look at it at a node level, which means that we look at, in this case, we will just combine these two genes in a normal sample node and a cancer sample node, and then we do differential expression. And then it again changes the picture, so we go to this one. See, now all the big nodes that contain a lot of different genes now just contains one gene. And as you can see here, these gene names have gotten a -B and then a number. That's because in the beginning, FunHub called it a box, and then it's basically a box with X numbers of genes in it. That's not really important. What's important is if you compare it to the original with only the one genes, you had this one was upregulated and this one was downregulated, and now they've actually changed. For this area up here, it hasn't changed because there is only one gene and nothing major is going to happen there. But as I explained before, there's a plausible explanation to why histidine is upregulated, and that is all thanks to FunHub. So histidine metabolism was a fairly simple pathway to work with. It's not very big. It has only a few nodes, which actually contains more than one gene. And it also has a metabolite we actually know how to work with. Another one of these metabolites that we know something about is the glycerophosphocholone, which is found here in the glycerophospholipid metabolism. And so basically, this is a fairly large network, and we're not going to use all of it. We only need this part here and down here. So we're just going to extract the information that we need. This is because it actually shows the final steps towards glycerophosphocholone, and it also shows some of the ways of FunHub and how it works, because it has some of the really large nodes, which contain like 21 genes. And that's really useful because it shows how wrong it can be if you interpret just this basic pathway like this, and if you don't use something like FunHub to show all the genes within a node. So if we do the same as we did with histidine metabolism, we download this pathway and we load it into Cytoscape, and we do similar coloring based on differential expression. It looks like this. And the metabolite we're going to focus on here is this glycerophosphocholone, which we know is upregulated in prostate cancer. And so is there a way to determine why it is upregulated? Well, if we start from down here, we follow the arrows, well, it's upregulated up here, but from there on, this road is not significant, this is not significant, and then it is upregulated, but then again this one turns the flow back that way. So there's nothing here that really says anything about why we should have more glycerophosphocholone. So then again, is there anything we can do with FunHub which changes this? Of course there is. And also this one shows how complicated this can become, because look at this node here which is not significant. If we run this through FunHop, we end up with this. And this is one of the larger nodes. It has 21 different genes, some upregulated, some downregulated. And this can possibly change the picture a lot. So we see some of these here. They have a fairly strong green, but then these two are very downregulated. We see up here it's the same. It's two very downregulated, two very upregulated. It's not as easy to get a proper view on this and notice anything about what's going on. So if we do just the same as before and apply the read counts instead, we get this. And just as before, we see that there are two of these that are fairly higher than the other ones. This is just a bit higher. It's down here on the light blue area, whilst this one here, it has a very high count compared to the other ones. We also here see that in these three nodes here, there's one gene that's the dominant one when it comes to read counts. And once again, we can say that since these are functional homologs, they will probably use the same rate, and the amount of the gene available will be the one determining how much you'll get and how the flow in the system will be. So if we compare this massive box here to the same one just using differential expression, we can see here that both of these two with the high counts are upregulated. And that is a very different picture than this single one, which is, it's not even not significant, it's not even found in our database or in the data set that we use. It's not found, so we don't know anything about it. So if we once again just combine all the read counts within a node and then do differential expression on a node base instead of a gene base and apply this to the network, we go back to the small one and we get this. And suddenly there is actually a possible explanation from here, because this one is upregulated and it goes through this one, which is upregulated, which stops here. This one's downregulated, so it ends here. So what we can say that we know that the count of this one is very, very, very high. This one has a fairly high number, but it's not close to the one down here. Not that we can compare these two, because these two are not functional homologs, but it still says a bit about the dynamics within this little system. So from not being able to explain anything on why there's higher amounts of 0-phosphocholine, we now actually have an explanation that is at least possible, and you can then work further on this, investigating and figuring out if this is the actual case. So once again, FunHop changed the picture. We learned something new. We have a plausible way of why this happened, which we didn't have before. And that's awesome. And just to make sure that it wasn't just us believing that this was actually real, we looked at the log2 reads from both TCGA and from a data set from the Princeton group. And just to compare them, and you can see here that it's pretty much the same. That they are on the line here means that they're very similar. And we also looked at the ratio within a node, because as I said, a lot of these nodes has one dominant gene, which is possibly the one responsible for the overall flow in that node. And this is from the big node with 21 genes, as you saw previously. We have the data both from Princeton and TCGA. The one from Princeton is a smaller data set, The Cancer Genome Atlas is, of course, very big, but the general overall tendencies are the same. It is this gene here, which has the highest amount, and it's the highest piece of the ratios, whilst all the other ones remain small, which means that we have some consistency in our analysis and we're not totally crazy. So, what have we learned today? Well, hopefully we have learned that networks can be very useful and fun and complex and beautiful. There's a lot of beauty in networks. If you remember the ones I showed you in the beginning on different diseases and stuff, I think it's really beautiful with all the different colors and shapes, and some of the groups who worked a lot with this, they do amazing things, and we can only hope to be part of that one day. We've also learned that there are many databases of all kinds of both data and biological networks. There are of course databases on a whole bunch of other things, apart from both metabolism and biological networks. You can find all kinds of weird data sets. We've learned that adjusting nodes and links can show different properties, like adding shapes and colors and thickness, and there are of course other ways you can do this by grouping them in different ways, and you can have different lines, different colors, there are so many different things you can do here. It's basically just your imagination, and it's not that difficult to tamper with the XML files that you use, so just be creative and do whatever you want. Could be fun. We've shown that a network can show a still picture of the biological situation. The gene expression data that we use is of course a still picture. It's not dynamic. It is possible to apply this to more dynamic data in many ways actually, but for us this is a still picture of the biological situation. We have seen that the picture is often wrong, but FunUp can fix it, and it works really well. If you want to look more into FunUp, you can find it on my GitHub. It will hopefully become a proper Cytoscape app one day, but I cannot promise when that will happen, but it's functional and you can use it from my GitHub. It's not a problem at all. I'd like to give thanks to my supervisors Morten and Finn for participating in this work. I'd also like to thank the MCC International Webinar Series organizers for putting all this together and of course inviting me to participate. It's been a lot of fun, and I believe there's a lot more fun to come. And most importantly, I want to thank you for watching my talk. I really appreciate it. I'll just leave here with some network humor, and I hope you'll ask any questions you might have in the comment section, and enjoy the rest of your day. Thank you.
On behalf of Mabalacat City College's Vice President for External Affairs, Mr. Raymond John D. Vergara, and the President of the College, Ms. Michelle Aguilar-Ong, we would like to thank Ms. Jers Teresa, our resource speaker for today, for sharing her time and expertise for our webinar episode on bioinformatics. Again, thank you, Ms. Risa. And for the last part of our episode on bioinformatics for today, we ask our registered participants to please answer the online evaluation and feedback form within today, October 10, to receive your e-certificate. The form will be closed at 12 midnight of October 11. Thank you and see you again next time.